one more announcement. If the technology works as I anticipate, you should receive an email on behalf of Alonso, second name I forget. He's the guy who's doing the social survey of how we form these interdisciplinary collaborations. So you hopefully will get a link to a questionnaire that he would like you to complete. It's entirely up to you whether you complete it, but clearly that would help his research. Yes, Tandy?
I've already completed it, and it was really easy to do.
Good, thank you. So that's a huge relief that actually the technology works. Great, thank you very much, Tandy. So yes, please do that if you feel so inclined. Right, so we now move on to the second session for today, and our first speaker is Rosalind Allen from the University of Edinburgh. Now, Rosalind is actually one of the organisers for another Newton Institute programme, which I think starts in December, and it's for 3 months, isn't it?
I've got a slide about it.
Oh yeah, okay.
Here it is.
Oh, there it is. So that's the ad for it, which is very closely related to this. Obviously, with a different sort of angle on it, but we're still talking about microbes. And I thought it would be very good to have Rosalind come here and sort of tell her story. And now she's probably slightly outside her comfort zone in this field of metagenomics, which is good because that's the whole theme of the week. So I'm very pleased about that. And I think this will really help, I think, what I'm hoping that this kind of presentation will help us to think about what are we doing all this for. Rosalind may well be, and her and the group of people that she works with may well be consumers of metagenomic sequence data. So we need to make sure that we can target that area. So that's the background to this. So let me just say a few words about Rosalind. She's currently a Royal Society University Research Fellow and proleptic reader in the School of Physics and Astronomy at Edinburgh. So quite a different sort of background to any of us. And if you want to know what proleptic means, you have to ask Rosalind. It's not as bad as it sounds. Her interest is in biological and soft condensed matter physics through theory and experiment. So it's a bit like Eddie. You know, he has one side on the sort of computational side and the other foot in the lab. So I think there's a similar kind of combination of skills there. But her PhD was in chemistry, so she's really spanned a huge range of disciplines, and so this is yet another challenge. And so I just hand over to you. Thank you.
Well, thanks for that introduction, which was longer than I've ever had before. So I guess one of my main purposes here is to advertise this programme, which, as you heard, is going on later in the year. And so obviously it's got quite a close connection to this programme. The difference being, I guess, that rather than focusing on sequencing specific, the purpose of our programme is to try to understand the kind of fundamental things that are going on in microbial communities from partly a theoretical perspective, but also experimental. So here's a list of the various workshops that are forming part of this programme, and if any of you want to come to any of them, that would be great. You can also find it on the web, obviously. So I guess the sort of motivation of that programme and also of my research is to try to understand what's going on in microbial communities. So I don't need to tell you that microbial communities are complex and diverse, and that sequence data has revolutionised our understanding of them. But I guess what's still missing is a kind of understanding of what are the fundamental processes that really drive the way that these communities assemble and function. So you can imagine lots of things that could be going on. Obviously there are metabolic interactions which are complicated, but there's also a whole community of people who work on how microbes cooperate. So for example, some microbes secrete products that help others, and others secrete toxins which are antagonistic to others, and so that somehow has to fit in as well. Sorry. I've got a bit of a cold. That has to fit in as well into how these communities work. But then you've also got viruses which prey on different types of microbes, and there's a whole school of thought which says that it's really viral predation which is responsible for microbial diversity. Then you've got people who are interested in small populations and demographic noise, and you've got other people who are interested in spatial structure. So obviously within these microbial mats, for example, you have a complicated gradients of chemicals and different species. And then other people who are interested in interactions with the environment and also how these things disperse. And so all of these things are probably important, but it's not really clear which of them are important in different circumstances and what their effects are on community structure and function. And so those are the things that we're trying to work on. And we kind of believe that studying these things in the natural environment is useful, but it's not the only thing you can do. And what you can do, maybe to understand things better, is to develop laboratory model systems which you can really control, and also theoretical models. And so in this talk I'll try and give you a bit of a flavour of what kind of thing we're trying to do. So the talk's got two parts. The first part is about our experiments, and the second part is about some attempts we make to do theoretical models. So the experiments are mainly thanks to my collaborator Andrew Free, who's from our biology department in the University of Edinburgh, and also the work of a postdoc Eulin Pageling and our long-suffering lab technician Fiona Strath-Dee. So this is all about nutrient cycling microbial ecosystems, and so microbial metabolism is really what drives the Earth's chemistry. So this is a picture, this is a schematic diagram of all the most important chemical reactions which really drive the way that the planet works. And you can't really see, but there's supposed to be a blue box here, and inside the blue box all these reactions are mediated by microbial metabolism. And so unlike human beings and plants, which basically all do the same chemistry, we all eat glucose and make energy, microbes can do all kinds of different things. So some microbes eat manganese and some of them eat iron and so on. And the result of that is that the Earth's chemistry can happen. So it's kind of important. We're particularly interested in the sulfur cycle. So this is a picture I stole off the web of the sulfur cycle. And the sulfur cycle, basically the important thing about it is that it cycles sulfur between its oxidized form, which is sulfate here, and its reduced form, which is sulfide. And so you see that in the soil you have cycling between sulfide and sulfate, and also in the ocean you have sulfate in the ocean and sulfide in the sediment. And this is important not just for itself, but also because it actually drives the global carbon cycle. Because you see this step here, what this is decomposition of organic matter. And it's believed that sulfur cycling microbes actually are responsible for about 50% of the Earth's decomposition of organic matter, because the products of this decomposition actually feed into this reaction, and so it pulls it along thermodynamically. So the important thing to say about this is that the steps which I drew in circles here are actually mediated by microbes. So you have sulfur reducing microbes which reduce sulfate to sulfide, sulfur oxidizers which do the opposite reaction, and decomposing microbes which actually do this step, so they decompose organic matter. And so microbes are really important in the sulfur cycle. But the thing is, and questions that you might want to ask are, in particular, how does this thing respond to perturbations? So, if the global climate changes, how much is the sulfur cycle going to respond? How is it going to respond, and how much? And also you might ask a hypothetical question. So, if we colonize the planet Mars, how will the geochemical cycles become established as a new territory gets colonized? But obviously you can't do well-controlled experiments on the global biosphere, and so for that reason we're doing our experiments on a laboratory model system. So this is a picture of our experimental system. It's a small plastic tube containing pond sediment and water, and it's about this high typically. And it's actually called a Winogradsky column after this famous early microbiologist, Sergei Winogradsky, who invented it. So this is actually a sort of famous system, but as far as we know, it hasn't been studied really in detail before, and that's what we're trying to do. So what you do is you get your plastic tube and you fill it up with pond sediment and water from the local pond, mixed up with a carbon source, which is cellulose, and a sulfur source, which is sulfate. And if you incubate this thing for several months with light, it actually develops a self-sustaining, nutrient-cycling microbial ecosystem. So inside the plastic tube, you get all kinds of beautiful visual transformations that happen. You get these vertically stratified purple bands and green bands and brown bands. It's very beautiful. And it actually sustains itself for years and years without any input except light. So it's kind of interest. And we're interested in it because with these systems we can easily apply perturbations, so we can vary the amount of sulfate, the amount of cellulose, and also the amount of light, for example. We can do interesting experiments where we take the community from one microcosm and transplant it into another. We can do all kinds of colonization experiments. And importantly, we can track the chemical gradients and the species composition, as I'm going to explain. So what's actually going on in this microcosm? As I said, it's a model for sulfur cycling. So actually what we have is at the top of the microcosm, where it's more aerobic, we have sulfur oxidizing microbes which transform sulfide into sulfate, and then that diffuses down, and at the bottom we have reducers which transform sulfate into sulfide, and that's coupled with the carbon cycle as I explained before. And so what's kind of interesting about this system is that even though it's simple and it's in the lab and we can control it, it does contain a lot of diversity. So if you do a sequencing on this, what you find out is, as you might expect, there are thousands and thousands of microbial taxa. So it retains much of the complexity of a real natural ecosystem. Just briefly, here's how we measure stuff. So measuring sulfide, it turns out that there are sophisticated ways of measuring sulfide profiles, which we didn't know when we started this experiment. But this guy in our geoscience department has helped us a lot. And you can do that by a method called voltammetry. So you stick a thin electrode into the microcosm, and you gradually move it down. And at each height, what you do is you get a current voltage plot. So this is the current through the electrode, and this is the voltage. So you scan the voltage at each height. And this plot actually has peaks corresponding to different chemical species. So this peak here at 0.8 and minus 0.8 volts is actually sulfide. And by measuring the height of this peak, it tells you the concentration of sulfide. So you can get plots like this, which is depth in the microcosm and the amount of sulfide at that depth. And so you can clearly see that in these microcosms there definitely is sulfide, but in this one there isn't. And we can also measure oxygen and pH. Measuring community composition, so obviously you know more than me about this. So far we've used a rather unsophisticated method, which is called DGGE, which probably you've never even heard of. And the reason we use it is because it's cheap, essentially. And what you do in this method is you amplify the 16S ribosomal RNA fragments, and you actually run them on a gel. So the gel contains a denaturant such that different sequences stop at different places along the gel, and you get a fingerprint of the community. And so this would be the fingerprint of one of our plastic tubes, for example. And this is kind of a nice method because you can quantify the similarity between different communities and so on. What's not nice about it is it doesn't tell you which species is which. And so we're also obviously doing 16S sequencing, and eventually we'd like to do metagenomics, but as yet we haven't, partly because we think it's too expensive. Okay, so the kinds of, I'll try and give you a flavour of the kinds of questions that we've started to address here. We don't really have any answers, but never mind. So one question you can ask, which is kind of interesting, is how reproducible is this? So if you just set up 100 identical microcosms and wait for 2 months, are you going to get 100 identical communities? And the answer is no, it turns out. So here's just a sort of picture of 3 representative microcosms that we got by setting up identical replicas. And you can clearly see even from this DGG that they're not the same. They have significant species or taxa which are not in common. And we've also measured a measure of community function, and this is much simpler than the sulfide profile. In this one you just stick electrodes at the top and bottom and measure the redox potential across the community, and you can see it's different in 3 replicas. So what we discovered from that is that there's apparent stochastic variability in the development of these things. And what we also find out is it depends on the volume of the microcosm. So we've done small ones and large ones, and the small ones are variable but the large ones aren't. So it's kind of a challenge to understand why that is. I'm not going to pretend that I do. So another question that we've asked is how does this thing happen in time? So from the start, when you set up the experiment, what happens over the 2 months that you incubate the system? And it turns out quite a lot happens. So what's plotted here is the sulfide at a certain depth in the sediment as a function of time after we start the experiment. And these different colours represent different amounts of cellulose that we added to the system. And what you can see is that quite a lot happens. So actually, at the beginning of the experiment, the community establishes itself like crazy, and it eats up all the cellulose and produces loads of sulfide, which you detect. But at some point, there's a kind of crisis that happens where it runs out of cellulose, and therefore it can't drive the production of any more sulfide, and the thing kind of crashes down. And interestingly, that happens differently depending how much cellulose you put in the system. What's also interesting is it seems to be more variable in the ones with more cellulose, which is something that we're looking at. So we have a kind of hypothesis that this sulfide crisis might have something to do with the variability that we see. And one thing that we're going to try and do in future is look at different size microcosms and see whether it happens differently in different sized microcosms. But the thing which is maybe which we've looked at most so far is actually perturbations. So I mentioned at the start that it's kind of interesting to know how ecosystems respond to perturbations, for example climate change. Here we've tried to do that in a simple way by setting up many microcosms with different amounts of cellulose. So cellulose is a kind of proxy for, well, it is organic matter. And for example, in the Earth's ancient history, it's believed that mass extinction events, when they happen, what happens is that there's a sudden influx of organic matter into the ocean because of all the dead dinosaurs and so on. And so it's kind of interest to know how a system responds to a organic matter loading. And more pragmatically, this can also happen when you have pollution in a natural water body. So what we've done is set up microcosms with different amounts of cellulose and sulfide, and we've incubated them for 20 weeks, and then we're just looking at the final state. How sensitive is it to the amount of, in particular, cellulose? And so here's our result. So what we found out so far is if we plot the total amount of sulfide in the system as a function of the amount of cellulose that we put in, we actually find out something quite interesting, which is that there's a switch. So if you don't put in very much cellulose, you actually see no sulfide at all. And after you put in a critical amount, which in this case is 0.2 grams, you see that the system becomes sulfidic. So what we see here is that the system shifts into a sulfidic state at some kind of critical concentration of this driver, which in this case is cellulose. And so the question is why. In fact, the question not just for this slide, but for all the previous slides, is what's really going on. And that's the kind of question that we try to answer in this program that we're organising and also in our work. So that brings us on to trying to understand why. And the way that we've been trying to understand this is by theoretical models. So this is the work of my student Tim Bush. And so we've been attempting to make some theoretical models that might explain what are going on in these plastic tubes. So how to do that? There are several ways that you could do it. One way you could do it is to focus on microbial diversity. So you could say, well, I want to model these 500 sulfur-reducing species, and I want to understand why they coexist. And that's an interesting thing to do, but it's not what we've done. What we've done is try to understand the metabolic interactions that are going on. And so we lump together all the sulfur reducers as one population, and all the sulfur oxidizers as one population. And we say, well, what's the simplest model that we can come up with which is going to explain the dynamics of this system? And we believe that it might be something like this. So we're saying that there's going to be 4 important populations. There's photosynthesizers, which use light, and they generate oxygen. The oxygen is used by the sulfur oxidizers to oxidize sulfur. And then you've also got cellulose degraders, which degrade the cellulose, and they produce hydrogen, which is used by the sulfur reducers. And so in our minds, the essential structure of this thing is basically a cycle which is driven by inputs from both sides. And so obviously this is a simplification, but we'll just see how far we can get with it. So actually we then simplified the thing even more. So then we said, okay, well, let's just look at this cycle thing. So we're going to ignore these inputs, and we're just going to say there's 2 populations, the oxidizers and the reducers, and they're cross-feeding in the sense that each one is feeding off the other one's waste product. So if you just sort of write down some equations for that, it's not very difficult to do. So we say that each population has a time derivative, which this is its growth term. So the growth of the population depends on the amount of its substrate, in this case S1, in a saturating form. So if you have a small amount of the substrate, then growth will be linear in the substrate. But if you have too much of it, then it won't be able to increase its growth rate anymore. It saturates. This is a standard thing to do. We also say that the population can't exceed a certain size. So for example, there's only so many bugs you can fit into the microcosm. And that's what this term does. And then we've got a death term. And then you have equations for x2, and equations for s1, and equations for s2 along the same lines. And you just write all that down. And it turns out that you can solve for the steady state analytically for that simple model. And if you do that, then the interesting thing to plot is actually the amount of sulfide versus sulfate, so the fraction of S1 in the steady state, as a function of the driver, which in this case is actually the growth rates of these 2 species, because with this parameter we encapsulate the driving from the photosynthesizers and the cellulose degraders, which we took out of the model. So here we've plotted the amount of sulfide versus the relative growth rates of these 2 populations. And the interesting observation is that the thing actually does behave as a switch. So under some circumstances, you can have a situation where this thing doesn't respond to changes in the driving, and then all of a sudden, when it reaches a critical driving force, it actually responds very switch-like. So it suddenly switches into, in this case, an oxidized state. And you can plot that on a plot like this. So this is just V1 and V2, and the color tells you how reduced the system is. So we're predicting that this system can, in some circumstances, behave like a switch. It can do this ecosystem switching thing. The reason being that actually our system is like a tug of war. So you've got the sulfur oxidizers trying to pull it towards an oxidized state and the sulfur reducers trying to pull it towards a reduced state. And the way the system's set up, if the thing is saturated so that if one of them increases its pulling rate, the other one can't respond, then it will just fall over into the other state. And it turns out this is a concept which is already known in a completely different field, so that's interesting. So we also find that when we put in a more detailed model, so then we put in the 4 populations and we have 2 boxes for the water and the sediment, and we just solve the thing numerically and find the steady state in silico, as it were, we get the same phenomenon happening. So now instead of V1 and V2 on the plot, we've got light and cellulose. So the light is driving the input of oxygen via the photosynthesizers, and the cellulose is driving the production of hydrogen from the cellulose degraders. We get the same phenomenon. So the model's predicting that if we varied the amount of cellulose at a fixed amount of light, we ought to see a sudden switch in the ecosystem state between oxidized and reduced at a critical amount of cellulose, which here will be here. So in some sense, that's nice because the theoretical model seems to explain the data, because the data does indeed show a switch at a critical amount of cellulose. So we made some progress, but it turns out it's not that simple. So when we started talking to geochemists and so on, partly from our university and other places, they said, oh, well, you don't have any iron in your model, and iron matters. And turns out that iron does actually matter. The reason being that the sulfide can actually react with iron to make iron sulfide, and iron sulfide is insoluble and unreactive. So another thing that could be going on in this system is that for small amounts of cellulose, we produce some sulfide, but it's not enough to overcome the iron, and so all the sulfide reacts with the iron, and we don't see anything because our voltammetry doesn't detect iron sulfide. And here, it could be that we just happen to produce enough sulfide that we overwhelm the iron, and that's why we saw it. So I guess the message of this is that the system is kind of complicated, and more work is needed to actually understand what's really going on. So that kind of brings me to the end. So hopefully this has given you a bit of a flavour of the kind of things that we're interested in. So I guess what we can learn is we believe that sequencing is important and useful, but you also need more to really understand what's going on. You need model systems that you can control and really try to understand in detail. That's what we're trying to do. So we're trying to develop tractable systems and theoretical models, but obviously there's quite a long way to go, and so we're still working on it. And that's partly why we think we need this 4-month-long program. So thanks for your attention.
We have a bit of time for questions. Yes.
So you showed that this evolutionary process is not very reproducible in the beginning, right? But then you use this ODE system to detect what's going on. So how does that?
Yeah, it's a good question. I think I guess the saving grace is that when we do experiments in large microcosms, it is reproducible, and so those microcosms that Tim did his experiment on were larger. So these ones are actually large enough that they are pretty reproducible, whereas the ones that I showed the other data from were 15 mils, so they were smaller. But yeah, you're right that with this model we're never going to capture variability, and we actually have no clue where this variability comes from, so that would be a whole different project, which it would be nice to do.
Why is there production of hydrogen from degrading cellulose? And is this just because it's a carbon source, or would you also?
Well, I missed out the fermenters. So my belief is that what happens is that the cellulose degraders break down the cellulose into smaller chunks, and then the fermenters break that down, and they release either hydrogen or organic acids or acetate, and that's the thing that's used by the sulfur reducers. So although I called it hydrogen, it actually isn't just hydrogen, it's more complicated.
So you had 16S data, is that correct?
To some extent.
To some extent, yeah. I was wondering, did you structure that in time, or?
Not yet. So it's running currently.
Yeah, so when you say laboratory system, I was a bit surprised that you just take a little bucket of soil and start working with that. I mean, wouldn't it be more controllable if you actually knew?
It definitely would.
What you had.
Yes.
And then when I see those model little plastic bottles. Did you take one sample and then you diluted it, and they've all been seeded from the same thing?
Yeah, so what you do is you take one big pot and you stir in the cellulose and sulfate as best you can and homogenize it all. And you actually sieve it to make sure it doesn't have sticks and stones in it and so on. And then you divide it up into the replicates. So you do the best you can. You're right that it would be nice to have a system where you actually know what's in it. And some people have tried to do that with 2 or 3 species, and it is possible to get the thing to self-sustain for a while, but it doesn't work very well, and no one knows why. So our system, you have to try pretty hard to get it not to make a functional ecosystem. In fact, it almost always does, whatever you do to it. Whereas those systems almost always don't. And so there's obviously something which is not understood about what you need to put in to get the thing to be stable. And so what it would be really nice to do with our system is actually extract species from it and try and combine them together and see what you need to do to make them stable. But that's quite a big project that we haven't had time to try.
One way that people, I believe, are using to get some slightly similar communities is introduce an enrichment step along the way that would sort of go in the direction Daniel wants to go. So you take your community from soil, and you can never sample the same soil, right? So you're going to have issues with the aggregates in there. But if you actually isolate a community that is able to survive in that environment and enrich it for a while, in somewhat altered circumstances, what you'll typically see is a reduction of complexity. And then you can take that and reseed sterilized soil with that.
Yeah, that's interesting.
That's maybe an approach. And then you can certainly sequence that, and then that would probably take care of some of your variation that you're seeing.
Yeah, that's a good idea. Thanks.
So we have people at Argonne that are thinking about this. Welcome to chat.
I'm interested in linear cellulose degradation. So there's in the degradation of cellulose, one of the problems is that it's crystalline, and that there's and the current enzymes that are well known, they act mostly on these amorphic regions of cellulose. But there's been new enzymes discovered recently that use oxidative mechanisms to break down this crystalline part. So they use oxygen to break down the crystalline region. I was just wondering if this could be relevant for your
Yeah, definitely. I mean, I guess when we have sequencing data, maybe we're going to find out what type of cellulose degraders we actually have, perhaps.
There's just few instances of such enzymes characterized so far. So maybe you won't even find them if you sequence the data, but if you could test different kinds of substrates, so cellulose versus already amorphic cellulose,
we could definitely do that. That would be nice. Yeah, that's a good idea.
I might have missed it. Do all your samples come from the same pond?
Mostly. I mean, so everything I showed here does, yeah.
So if you go to a different pond, or you put soil and pond water, or I don't know, potting compost or anything, would the stability of the system be different? Do you know?
Well, we've done some of that. What we've actually done is some experiments where we took the same pond sediment, but we sterilised the microcosm, and then we inoculated it with communities from different lochs in Scotland. And we were interested in whether you get the same community at the end of the day or different ones. And what we found out is that you get different communities from different lochs, and you also get different communities from different replicates from the same loch. And the contribution to the difference between individual microcosms is about 50%. So it's 50% coming from the source and about 50% coming from this random stochasticity that we don't understand. So it's complicated.
I have a question of my own. This is clearly a rather special system you're studying because it is so robust, which makes you makes me ask the question, well, maybe this is not going to be very generalizable.
Could be. I think what would be really nice is to try the same thing with a different system. So what would be extremely nice is if there's a way of doing it with gut microflora. So my collaborator Andrew has found a paper where they have little cylinders where you can get gut microbes to kind of grow in it's sort of like a chemostat but much less high tech. And you can imagine doing a similar thing, which would be really nice.
I mean, you're running these things to stability. Stability seems to be a kind of a thing that you require. But I mean, I wonder why that is. I mean, if something would actually run for 2 months and then just fall over and everything dies, would it really matter? Because you could still study the dynamics.
You could, yeah. It would be a bit more difficult because the good thing about this is you can just ignore it for 20 weeks and then come back. And
I see.
but you could do it, yeah.
Yeah. So I mean, and you don't. I wasn't clear whether you actually sample through time.
Yeah, some experiments we do and some we don't.
Okay.
So we've got one where we do, and then Tim's thing, this one here, we didn't.
Okay. And so presumably then you can study whether or not these dynamical models that you set up, whether they really are describing what you actually see.
Yeah, that's what we want to do. Yeah.
Okay. Yeah, 'cause you know, you predict this sudden switch in some conditions.
Yeah. But you can go much further than that, and that's what we're going to do, hopefully.
And there must be some. If you're studying a system which is stable, it's a bit more like a natural system than
Yes.
so it's something which is just dying in the lab.
Yeah. But on the other hand, you know that this is not going to be really a natural system because in a natural system there's so many other players.
Yeah.
And so many other processes, and this system may be rather special in the sense that perhaps the self-recycling is the dominant thing that's happening. But
yeah, that's definitely true.
I would imagine that there's all kinds of other things going on, all kind of other processes. And is it really true that in any ecology there really is stability? Doesn't everything change all the time?
Well, who knows?
It's good one.
Yeah.
You want to be. you want to do something tractable.
Yeah. I think there's another question there. Yes.
Are you planning on taking any other kind of physical measurements, oxygen concentration, pH?
Yeah, we can do oxygen concentration. So that works with a different probe, but we can do it now. pH we can also do. I think what would be nice that we don't currently do is also gas production. So, for example, it's possible the systems are producing methane, and it would be nice to collect gas from the top and analyse it. So, I think that's quite easy to do, but we just haven't done it.
Are you also producing H2S?
Well, probably. It smells like you are. So, yes, I think so from the smell, but we haven't actually quantified it.
Well, you can do that with a lead acetate strip, paper strip.
Okay, well, that's a good thing. Okay.
And the other thing is, if you measure over time, at what point in time does your environment go anoxic?
Well, actually, I mean, you know that from the sulfide data, right? So it goes anoxic when the sulfide goes high, so around 8 weeks. And we also have oxygen data that shows the same thing.
And how does that correlate with the consumption of your carbon source, main carbon
We don't know
source?
that because we haven't measured carbon, which we should do, actually.
You mentioned that you have these sort of unexplained results. So the size of the system seems to make a difference. If it's bigger, it seems more stable.
Yeah.
Now, I don't know if this is relevant, but it's a well-known phenomenon in statistics that if you have a small sample, you have more variability just through randomness.
Yeah.
If you've got a large sample, well, the ups and the downs sort of tend to cancel each other out.
Yes.
There's a thing called central limit theorem
Yes.
to prove this kind of thing. Now, this is a very different system because you've got, it's one big system rather than lots of little ones.
Yeah.
But it does make you wonder, or at least makes me wonder, whether or not you've really got if you could almost imagine it as a sort of compartmented, if there's not too much of cycling of the microbes from one end of one side of the tube to the other. Maybe there is a vertical positioning, but if they tend to sort of stop where they are, you might effectively having a sort of weakly correlated spatial system, which could actually, which central limit theorems exist.
Yes.
So it could explain it. I don't know, but
I think this stochasticity thing is very fascinating, and it should originate, as you say, in some kind of statistical thing with small numbers. But where the small numbers are is a I mean, they could also be in the sample at the beginning. You could say, well, it's a rare species that you need to populate this thing, and when you sample it, there isn't very much of it, and that's where you're sitting. But we just don't know. So
maybe. Well, I guess you could study that by varying the amount of starting material.
Yes.
So you've got two ingredients. One is the amount of starting
Yes.
material. The other is the size of the tube that you put it all in.
So we did that actually, and for the archaea. So we were able to look at archaea and bacteria separately because they have different 16S genes. And so for the archaea, you do indeed see what you would expect, which is that when you dilute the inoculum, you get more variability. And for the bacteria, we didn't see that, which could be because we didn't dilute it enough, because there's more bacteria than archaea, or it could just be that something's wrong with our reasoning. So it's a mystery.
So I've got one more question. If you had the money to do sort of metagenomic sequencing, would this help, or
I think it would help, yeah. Well, I hope that what it would tell you is which functional groups you've got proliferating at different times, and that's exactly what we have in our theoretical model. So hopefully it would be a much more direct way of actually matching the experiment with the model. And hopefully what it would also tell you is within a given functional group, so like within sulfur reducers, how many species have you got, and what are they, and is it always the same ones? And that kind of thing would be very useful.
Comment at the back.
Yes.
I'm surprised. I was actually surprised earlier when you said 16S sequencing was expensive. One of the things I always ask is, I ask people, what do you think 100,000 16S reads will cost? And just to shortcut this, it's about $20 US.
Yeah, but we have hundreds and hundreds of samples. I mean, now we are now doing it, so okay. I think your argument is correct.
Okay.
This is a comment, it's not a question. Sulfur is very important. As we try to feed 9 billion people in the future, sulfur is a limiting factor in being able to grow crops just the way nitrogen is. So there's deep interest in trying to understand the role of microbes in making sulfur available to plants. And I'm at an institute that does sequencing for projects like this, the JGI. It has a user program, and you apply for access to it. And so I urge you to look, and and the Department of Energy that funds the institute is interested in, you wouldn't come there with a gut microbiome project, but you would come with this kind of project, and it provides metagenomic sequencing analysis. And right now there's a user program, and so I urge you and other people here to, if you have sequencing needs that can connect to energy and environmental issues, that you look at the JGI website that talks about its user program and think about applying for it. And if if people have questions, they could direct them to me.
Okay.
Okay, so I think we're done. Thank you very much, Rosalind.
Thanks.